Well, welcome everyone to the Alaska SeaLife Center. My name is Darren, and we're going to do a program today called The Scoop on Poop. So I'm going to talk to you a little bit about the SeaLife Center and some of the work that we do, and then we'll talk about these stellar sea lions that are swimming around behind me, and some of the research that we have going on with those animals out there in the wild, and I'll probably talk a little bit about what we have going on here in our facility as well. So first and foremost, our location. We are located right here in Seward, Alaska, and so we're right on the Gulf of Alaska. If you're not already aware, Alaska has some very, very important fisheries. The world's biggest fishery, in fact, is Alaska Pollock, and we catch about 3 billion pounds of pollock every year, and that's what's turned into fish sticks and fake crab, shipped all around the world. All of the fish that McDonald's sells is Alaska Pollock, from mostly out here in the Bering Sea between Alaska and Russia there. And of course, as the Arctic sea ice shrinks back and melts further every year, there's a lot of interest up in the Arctic Ocean. Currently, there are no fisheries up in that area, but there could be some potential up there in future years. So there's a lot of interest in these oceans around Alaska. And that's why we have the Alaska SeaLife Center, so that we can help everyone understand what's going on out there in those oceans and teach others about them. Make sure we do our best to take care of them. So this is just a short video of some of the animals and some of the facilities that we have. I'll fast forward here for just a second. These are our harbor seals. They're very cute. Got to start with the cute ones. And then we have the stellar sea lions. These are the largest sea lions in the world. That guy's name is Woody, and he weighs about 2,000 pounds. We'll see more of him in a little bit. And then we have a diving seabird tank, which is amazing. We have puffins and guillemots and murres, and you get to watch those birds fly underwater past fish and anemones and sea stars. Super cool tank. It's actually the deepest diving seabird habitat in North America. Of course, we've got lots of fish and other invertebrates as well. Some of the largest sea stars in the world live in the North Pacific here. The sunflower stars up to 3 feet across. And we've got a fun little touch pool where our visitors can put their hands into the 40-degree water and see what some of those invertebrates feel like. So that's a very popular exhibit. It's a great place for students to come. In fact, there was a group just last night spending the night here, 5th graders from Anchorage, which is just a little over 100 miles from here. And students come in quite a bit to enjoy our facility. We do have a rescue program, so we rescue injured and stranded animals. That was a little sea otter pup. This is a harbor seal pup. Both of them were orphaned. This harbor seal was able to be released back out into the wild, so there she goes back out into the ocean. And finally, we have our research program. So we have a team of several dozen scientists working through the Alaska SeaLife Center, and we also work closely with the University of Alaska in Fairbanks. And our scientists work here in our facility. We've got laboratories on site. We also have remote field sites and field equipment like remote camera systems that we can control by microwave transmission, so we can have live images of those animals out there. We even have a scientific dive team. So the dive team is specially equipped to go out and collect, observe, record data underwater. It's just another specialized training that you can get as part of a dive certification. Scientific diving is part of that. And of course, a lot of visitors come to Alaska just simply for the view, so I'll throw that in there at the end here. This is Resurrection Bay. It is a glacially carved fjord, so long, narrow, very steep-sided bay that was carved out by glaciers and filled in with seawater. And even though it's only a few miles across, that bay is over 900 feet deep at its deepest point. So those mountains are very steep, and they just keep on going down when they hit the water. So that is where we are in a nutshell. And we're going to talk about our stellar sea lions primarily today, and the research that we do with them. So let's talk first about just the animals in general, some of their natural history. These are the biggest sea lions in the world, like I mentioned. Males grow to be about 4 to 5 times the size of the females. So the females will be around 500 to 600 pounds, and the males will be up to about 2,500 or 2,600 pounds in the springtime during the breeding season. They bulk up. So this is a female that you see swimming by right there, and I'm actually not sure how many are in the habitat right now. Never know who's going to be in there at any given moment. All of these stellar sea lions are here on research permits, so their primary purpose for being at the Alaska SeaLife Center is to participate in research projects. But a side benefit is we put them in a big tank with windows so our visitors can come and see them. But I can never guarantee that the animals will be in the tanks because sometimes the researchers need to collect samples or move the animals around for various reasons to put some together and some not together. So I never know who's going to be in the tank. But I do have some recorded video of these animals. So let's take a look at some of the things that they are capable of. So this video will show you a little glimpse into Woody's life here at the Alaska SeaLife Center. So on land, these animals are, of course, built mostly for swimming, but they are capable of climbing around on land. They can stand up on all fours, and that's a characteristic of sea lions versus seals that have very short front flippers and have to lie on their bellies. Sea lions can get up on all fours and climb around on steep, rocky areas. So part of their habitat can include a very steep-sided rock face. They can dive into the water off of these higher rocks. They're capable of moving around on land very, very well. They are very social animals. They like to congregate out there on the rocks. And one rock in particular, one island in particular that we have a close eye on is the Chiswell Island Rookery. And this is what it looked like back in 1978. Probably around 200 animals in this image. And back then, we weren't too concerned about the population of stellar sea lions. There were a lot of them. But 20 years later, this is that exact same island, and this is a rookery. It's a place where they gather to give birth and raise their pups. So the fact that most of the animals are gone was very concerning to scientists around that time. So initially, they were listed as a threatened species, and then later it transitioned to endangered. In this area of Alaska, there are two separate populations of stellar sea lions. Their range goes all the way from Russia and Japan on across into the eastern North Pacific and down into as far as California they can be found. Most of them are found right around the Bering Sea and south central Alaska. But you can see there's a line there that says 144 degrees west. That line of longitude separates the eastern stock from the western stock. And the eastern stock was also listed as threatened on the endangered species list, but has since been delisted. Just in the last few years, that population was removed from that list because it had stabilized and recovered from the initial decline that started back in the 70s. The western stock, on the other hand, has not recovered. And so that's why we have those animals here at the Alaska SeaLife Center. They are still listed as an endangered species. And you see all those red triangles. Those are all rookery areas. And again, very important spots for these animals because that's where they raise their pups. So just a quick glimpse of them underwater, since there's not too many in the habitat right now. This is what they look like together. And there's going to be two females swimming together here, and then the big male will come out from under that rock on the left. So watch him come out and see how much larger his body is compared to theirs. He is almost 10 feet long and roughly 2,000 pounds, although in the springtime he'll be about 2,400 pounds, and in the fall he'll be about 1,700 pounds. So he loses all that weight every summer through the course of the breeding season. Of course, the food that they like to eat mostly is fish. And you'll see in this video, hold on, I got to turn the sound on for this one. It's way more fun with the sound. All right. Hopefully you can hear that. That's the sound that the stellar sea lions make.
Good. Wait. Wait.
Watch how he swallows this fish. This is salmon. It's about 2 feet long.
Good boy.
So that was a fish about the size of my arm, and he made it disappear like it was a chicken nugget. These animals obviously swallow their food whole, and Woody, when he's gaining weight in the springtime, will eat about 65 pounds of food a day. That's the most that he'll eat. So this is just a quick training session so you can see some of the behaviors that he's trained for. And again, he's here for research. He's not a show animal. We do let people watch when he's doing these behaviors, and it's fun. But most of these behaviors are related to making sure that he's got a good, healthy life here at the Alaska SeaLife Center. So when he lifts up his flipper, it's not because we want him to wave to the crowd. It's because we need to see under his flipper, and we can't just pick him up. And teaching him to roar on cue like this helps us see inside his mouth, so we can look in, check his teeth and gums, and we also brush his teeth. But the trainers always work with him in protected contact. So you can see the trainers are not in there with him. They are never in direct contact with him. There's always some barrier between them.
Right here.
There we go. All right, so, and the reason for the protected contact with that particular animal is he's just so big that even if he just slipped, he could easily crush a person. And the sea lions, the males in particular, are very territorial, especially during that breeding season. So he doesn't want anybody in his space, but he works very, very cooperatively through the bars. We can brush his teeth, get right in there, and he does a great job. He's an excellent animal, very well trained. But again, they have to have some protection for those trainers working across through the fence, so to speak. All right, so that's a little bit about Steller sea lions. And let's look back at their population. This is going to be one example of some data that were collected at a rookery called Marmot Island. And looking at this data, this plot, if you can't see the numbers at the bottom, this is 1950, 60, 70, 80, 90, 2000, 2010 would be the latest data. What would be some information you could gather from this chart?
There's an obvious population decline in the 1990s.
There is an obvious population decline. When did it start?
1985, looks like.
Around 1985 was this precipitous decline. Now, we've got some weird data back here. What was happening back in these days?
I would guess that at that time there wasn't a lot of data being collected, and so it's sporadic.
Sharp. All right, exactly. We don't really know a lot about what was happening back here because we weren't concerned. So there are a few counts, but they're from way back in 1957 probably. Just a couple of random numbers. So we really can't say a whole lot about here, but clearly there was a big population, several counts, got over 8,000 numbers, almost to 10,000 at one point. And then when scientists started to get concerned, you could see a very, very sharp decline. So this was clearly a trend. And then it bottomed out down here. So they didn't disappear entirely, luckily. But we've seen some just very small signs of recovery so far. That's why they're still listed as endangered. There used to be a very large population. Over 80% of them overall from this whole population have disappeared. And we've seen small increases in the last 5 to 10 years, but they are definitely not out of the woods yet. So once we noticed that there was a steep decline happening, that's when we started doing very regular counts. And of course, once animals get listed on the endangered species list, they get a whole lot of extra attention and a lot more research dollars put toward them. And so that's one of the reasons why we have them here at the Alaska SeaLife Center. There's not as much research funding being put towards Steller sea lions as there used to be, but there is still some research and definitely ongoing monitoring. The current programs that we have with our animals are right now mostly focused on maternal investment and what kind of energy requirements there are for the mom to raise the pup, because she will be out on the rookery ready to give birth right at the end of May and the beginning of June. And she'll give birth to a pup, begin nursing it, but after about 1 to 2 weeks, she will breed with a male and then potentially become pregnant again, and will be pregnant again for a full 12 months. There's a 9-month gestation period, but there's something called embryonic diapause. And so the embryo begins to develop a little bit and implants in the uterine wall and then stops and goes into this diapause state. And then around October, the female's body basically decides to proceed or not, and will reabsorb that embryo if it's not going to proceed with the pregnancy. But if all goes well, she'll be nursing that one pup for a full 12 months, and give birth to another pup the next year. So there's a huge energy demand on that female to be nursing this fat, blubbery sea lion as it grows in its first year, and at the same time growing another fetus inside her body. So what we're doing is focusing a lot on studying their hormone levels and determining when they go into estrus and when is the best time to put them together with the male and have successful breeding here in an aquarium setting like this, but also tracking the development through time of that female and looking at how much energy she has to put out to raise that pup and be nursing while she's pregnant again. So this year we are hoping for successful breeding. We have had 2 pups born already from Woody, but Woody is now retired, and so we have his successor, Pilot. I don't see Pilot in the tank right now. He's a bit bigger than these females, but he's not nearly as big as Woody is. He's about half Woody's size, but he will be 5 this year and should be capable of successful breeding. So we've got our fingers crossed that next year we'll have some more pups and we'll be able to continue studying. But in the meantime, we are very focused on studying the hormone levels of those females, and we'll get back to that info later. So tell me some of your hypotheses. Knowing what you know so far about stellar sea lions, what do you think could have caused this population decline?
I'm not really sure when we started like drilling for oil up there, but that definitely could be a big possibility. That like mercury levels definitely increased since the 80s, so that could definitely be a part. Maybe biomagnification with all the fish.
Very good. So mercury is just one example of a pollutant, but biomagnification is also critical. We're talking about pretty high level predators. They are not the very top. They do have one main predator that we're very sure of here in Alaska, and there might be one other predator that eats these animals besides humans. There are Alaska natives who are allowed to hunt stellar sea lions, but pollution and biomagnification is definitely a concern, and we'll collect samples using little biopsy darts. We actually have a crossbow set up with a little tiny dart that takes a biopsy punch from a sea lion, so we get about 10 to 20 meters away to get an accurate shot on that animal, and shoot the dart in and just retrieve the dart, and it's got a little biopsy punch that has gone through this hair and skin and blubber layer, which is anywhere from like a ½ inch to 2 inches on these animals, depending on the time of year and health of the animal. And then right down to the edge of the muscle, and we can sample those various tissues to look at contaminants like mercury, PCBs, dioxin. So we've been keeping an eye on those pollutants, but it doesn't seem like there's a significant problem with chemicals like that. But it's certainly something that scientists keep a watch on just to make sure. What's another hypothesis?
I would say that you know how we were talking about the fisheries before. I mean, if we're over harvesting up there, I mean that's definitely going to leave them with the lack of food source. Yep,
definitely a potential problem of just being out-competed by the fisheries. Our fisheries are pretty efficient. And so when these animals were listed as threatened and then endangered, there were certain areas that were just off limits for everything. No boats can go there at all. Certain protected rookeries. They designate critical habitat for endangered species as part of a species recovery plan. So there were certain protections put in place. But then there are also quite a few studies done on what these animals do eat, and a significant effort put towards just making sure that those fisheries are not out competing those animals and taking too much. So we might find evidence of that as well. What else?
Could changes in the temperature of the ocean have any effect? Like maybe it's getting too warm for them?
That's a great question. So we know that the climate is changing. And in Alaska and the Arctic in particular, things are warming up faster than most other areas, partly due to, if you guys are familiar with the albedo effect, as that ice and snow melts away, then instead of having a big white reflective surface up here, you have dark ocean or land that's going to absorb a whole lot more energy from that sunshine. And so that sort of compounds itself and causes the temperature to increase even faster than other areas of the planet. So we've definitely seen some warming. But when you look at just the sea surface temperature, that's like 1 degree Celsius, and that amount of change is not enough to really impact the stellar sea lions directly. It could make them change their energy demands somewhat, but they have pretty effective means of heating and cooling because they can haul out on land, and like those big bulls, they haul out in early May, get out onto the Rookery, and they just sit there for 2 or 3 months. They're out of the water. So they have ways to cool themselves down because although it doesn't exactly get hot here in this area of Alaska, it can be a calm, sunny day, and their body can overheat pretty quickly. But they have ways to just move a lot of blood to the outer surface of like their flippers, and they can get a flipper wet and hold it up in the air. They can cool themselves down. So a slight change in the temperature is probably not going to affect them directly, but it can affect their food source directly because, of course, they feed on cold-blooded animals like these fish over here. And cold-blooded fish may have a very narrow range of temperature where they can survive. And so if the stellar sea lions are focused on a particular prey type and that prey type moves on or dies because the temperature changed in the water, then the stellar sea lions could be in serious trouble. So just a simple change in the temperature, it's not likely to affect the stellar sea lions directly, but could definitely impact their prey source. All right, what else? Something else that I mentioned that you guys haven't jumped on.
Did you already say the pollution may be causing infertility?
Oh, that's a good one. So reproductive success is a great, just kind of general thing. And that's often an early indicator of things like stress. And stress could be caused by a lot of different things. If it's a pollutant stress on their bodies, they might have lower reproductive success. And so this is a really poor image right now. This is from our live cameras. But I'm showing you this because we've been watching these animals using these remote cameras since 1998. And we can identify individual sea lions out there. And so we know that if that same female comes back year after year, we can see if she's still got last year's pup with her. We can see if her pup survived, and we can see how many pups are born each year. So we've gotten consistently, gradually more pups born each time. And this past year was actually, in 2014, was the highest number that we've ever seen in the last 20 years or so, out there at that particular rookery. So we can see their birth rates are actually looking pretty good. What we don't know is what's happening to those young animals. Why are they not growing up to become adults? So that's something we've been able to learn through those remote cameras and other monitoring methods that they have. They do flyovers and counts from boats sometimes. They have mark recapture studies where it's not really recapturing them, but recite. They put brands on the animals as pups, and so you can identify them even from when they're just born. You can identify them throughout their entire lives, so they use branding for that. So we have means of seeing these animals and how well they're reproducing. So there doesn't seem to be a direct impact on their just straight up reproductive rates. But that is a good thought. How about something that might be eating them? What sort of predators do you think they might have?
I thought you said that they really only had one predator, sir, being the orcas.
It would be orcas, yep. So orcas are the main predator of the stellar sea lions, as far as we know. What we don't know is exactly how many sea lions does an orca need to eat. We know how many orcas are out there that might be eating these sea lions because it's a very small population of orcas that actually eat marine mammals in this area of the North Pacific. Most of the orcas here feed on fish, and that's it. But we've definitely identified a population of orcas. They're called the transient orcas that do feed on other marine mammals, and some of them prey on stellar sea lions. What we don't know is how many. So one neat study that we've done is by putting in what we call life history transmitters. We capture stellar sea lions, we anesthetize them, we do a surgery, and we implant a tag it's a satellite transmitter, but it's programmed to record temperature, and these are the earlier versions. They recorded temperature, light levels, and salinity. And so on the inside of a sea lion's body, none of those should really change very much. Temperature, salinity, and light level, it's just dark. But when the animal's body decomposes or is ripped open somehow, all of those things should change. And if it's ripped open by something like an orca, it would change very fast. Then the tag is programmed to, it floats to the surface and it's programmed to transmit its data to a satellite. So we can see then, did the temperature go straight down because the animal's body was torn in half, or did it decrease gradually over time because it starved to death and then sort of slowly decomposed on the beach? They can tease out those differences. And from the early stages of that study, we saw, I believe it was 11 of 12 animals that appeared to be taken by predators. There is some question, though, whether all of those predators were orcas. There's a shark called the Pacific sleeper shark that may be preying on stellar sea lions, but we don't know that much about them yet. So we're trying to do more focused studies to see if those sleeper sharks are actually preying on stellar sea lions. It's a difficult thing to be able to pick out from the kind of minimal sparse data that we can collect. You know, going and capturing an endangered species and implanting something in its body is pretty invasive. So we can't just do that to, 500 animals. We have a pretty small sample size, but we have enough information to be pretty suspicious of those Pacific sleeper sharks. And they can get to be 25 feet long. So we know they're big enough. It's just a question of whether they're doing it. And there's also humans that can hunt stellar sea lions legally. The Alaska natives don't hunt very many stellar sea lions. They do hunt them from time to time, but it's not a primary food source or anything. So that's a pretty small number. And then, of course, there's the possibility of illegal poaching or animals getting killed through the fishery, not just competing with them, but actually catching them in nets. Some of those are recorded. They're all supposed to be recorded, of course, but some of those may go unnoticed. So there's a lot of potential reasons why these animals are declining. But what we're doing today, looking at their scat, of course, is going to help tell us what they're eating and if their prey base has changed somehow. So we've been looking at stellar sea lion scat samples on and off for many years now, and it helps us take a good look at what they've been eating. It's not an exact science, but it's a great way that's non-invasive to go and determine some of the food items that they've been eating. So I'm going to show you, or we're going to look through these scat samples and see some of the information that our scientists can glean from those scat samples. We are not experts today, but we can learn quite a bit, even being totally uneducated on the subject. So we have a little ID chart that you guys have, and you've got your scat samples, and you have your worksheet. So the worksheet is just a way to kind of guide you through the process of sorting the sample and then finding some of the data. So you can go ahead and dump out your samples. I don't know if you guys have trays to work on, or even just a piece of paper is good to work on, and then you can pour your sample back in when you're done. I see some of you do have, it's like you have gloves on. Just so you know, these samples are only from our resident animals. So there's not going to be some unknown virus in there, but it did go through the body of a sea lion. So you definitely want to wash your hands afterwards, but I don't know what your regulations are. I don't wear gloves with these particular samples, but the scientists, when they work with wild animals, always have to, because you never know what kind of diseases the animals out there might have. And some of them are able to move across species. Those are called the zoonotic diseases, ones that can infect multiple species, animals and then humans as well. So the first step is just to sort your scat sample into piles of things that are similar. So it's usually easiest to start with things that really jump out at you. Pick out the really obvious things. And as you're sorting, if you want to compare to the ID chart, you can, or you can just focus on sorting. Feel free to ask questions as you go. If you find something you're not sure of, or if you have a question about the stellar sea lions in general, just toss them out there. I'm going to be sorting away here, but I'll shut up for a minute, and if you guys have any questions, throw them out.
Yeah, I got one. Do they have any known migratory routes, or do they have specific route that they tend to swim each place? Because obviously they have that breeding place, but do they have
yeah, that's a good question. They do. They move around a bit, but they don't tend to have a huge home range. And they have they'll have haul outs that they use only in the summertime or the winter time. Then they tend to get to more protected areas in the winter, at least a bit more protected. But they do tend to come back to the same rookery where they were born, at least many of them do. So they're kind of homebodies, but they do have the ability to swim a long way. They just don't move from one place to another far distant place and then back. They tend to just kind of stick in a relatively small home range and move into more protected areas in the winter, and then they can move out to the outlying areas in the summertime. There are also specific areas like the Chiswell Island rookery that I showed you a picture of. That's only used in the summertime, and during the breeding season, it's only going to be the big bulls that are actively breeding, and females are always welcome. The bulls aren't going to chase them off, but young animals are not going to be allowed out there during the breeding season. The big bulls will chase them off. So you won't see a bunch of juveniles until at the end of the breeding season when everybody's just sort of tired and lazy. Then you'll see a big mixed population out there. So those younger animals, they'll be confined to more of the outlying areas. Sort of the younger social crowd will hang out there when they're not quite ready to breed yet. So they do have different areas, but not long distance migrations.
And is that breeding site? Is that a protected area?
That one doesn't have any particularly special protection, but with any marine mammals, the laws are a little bit open to interpretation, but really, strictly speaking, you are not allowed to harass any marine mammal, but harassment can be anything that changes that animal's behavior. So if it looks up at you, technically you've just broken the law. So they recommend you stay at least 100 yards from any marine mammals. But if the marine mammal swims up to you, you haven't done anything wrong, as long as you didn't approach it to within less than 100 yards. How enforceable that is a little bit tough. So that particular rookery is actually, it's about 35 miles from here. And I would say some of the tour boats and fishing boats give it more respect than others. And that's something that we see from our remote cameras, is we see when some boats get too close. Now, it's not the kind of thing that we're going to call up law enforcement and have them busted, unless we saw something really egregious. But it is something we can make a note of. And we've had some studies with harbor seals where we watched kayaks and tour boats coming by, and we got a much more scientific handle on what kind of vessels and what kind of behaviors disturbed those animals more. And we were able to teach the kayak guides, the local folks, what we learned, and then we saw a lot less disturbance. So that's not something that we're super concerned about with this particular rookery. At least I've never heard the scientists voice much concern. But it doesn't have any extra protection necessarily. There is one farther out that is totally protected. I think it's a 3 nautical mile zone that no one is allowed into. How's your sorting coming? So far, so good.
Can you repeat?
How's the sorting process? Is it making sense just to get things into different piles? And have you been able to identify anything yet?
I haven't identified anything yet, but it's, I don't know. I find it fun to separate them. It doesn't exist.
It's very satisfying, isn't it? So I would encourage you then to go on into step 2 on your worksheet. This worksheet's more intended for like middle and high school, but take a look at your ID chart and see if there's some things you can identify. There are definitely some parts that are not shown on your chart, but I'll zoom in on some of my parts too and help you identify some things. But again, we're not experts. We, even here at the Alaska SeaLife Center, we don't have anyone who's specifically an expert on identifying all of these parts. So there's probably around a dozen things that I could identify in here. Beyond that, we send these samples out to experts. But because these samples are all from our resident animals, I know the different types of fish that we feed them and other things. So I can identify a little bit more than what I'd be able to do. If this was a sample from a wild animal, I would really not even be able to guess at what kind of species they've been encountering out there in the ocean. So what sort of things have you been able to identify yet?
Well, I found a bunch of like operical, like gill covers, pretty much. I have.
Oh, the big flat bones. Okay.
Looks like I've got about two vomer bones so far. It looks like I got like a thoracic vertebra.
Are you an expert? You sound really confident.
Well,
have you done a lot of fish dissections? Those were parts that I can't really identify, but we did have some scat samples recently that had some really big identifiable fish parts. And I don't have any in my sample here, but you may have gotten some of those. So very cool. How about anybody else? Anything you could identify?
I think I have walleye, pollock,
a walleye, pollock, otolith. Yeah. Those should be fairly common in your samples. And now these, again, are from the Alaska SeaLife Center's animal population, but you probably should have all found at least one of these. Pollock is, I think by mass, probably the biggest thing that we feed here. And again, that's the biggest fishery in the world for human consumption. Can't see if I got a pollock. I'm looking in my bird habitat here. They're around, but it looks like they're all hiding right now. Pollock are a type of cod, and we know through studies like these scat collections that stellar sea lions in the wild do indeed eat pollock. And that was one of the big initial concerns back in the 70s, because there had been what's called a regime shift out in the Gulf of Alaska. In certain areas, there used to be a lot of herring as a prey base, and that was just sort of the bulk of the fish population out there. And that started to transition over into more pollock. And the initial thought was, well, herring are a really high-fat fish, they're very oily, and a super high-quality food source for these stellar sea lions. Well, if they're not getting as much herring, now they're transitioning to pollock. It was called the junk food hypothesis. Like, pollock just wasn't good enough for them. But we could show through studies here at the Alaska SeaLife Center that just feeding pollock to these sea lions was actually okay. They could thrive on that type of fish. That didn't necessarily answer the question of how much energy it takes for them to dive down and get those pollock versus catching herring. You know, it's certainly possible that herring were in a more accessible location, or the pollock were deeper or more far-flung, and it was just harder for them to get them. But pollock are a bigger fish than herring. And just through caloric intake, it appeared that pollock was a good enough food source for them. So we know that pollock are important to these stellar sea lions, which is a big deal. Here we've got the biggest fishery in the world that we're catching out there, and then these endangered species that are also interested in eating those animals. So we need to be careful not to accidentally catch the sea lions in the nets, for one thing, but also to make sure that the sea lions have plenty of fish out there to eat too. So Alaska is very proactive in managing its fisheries and trying to make sure that everything is done in a sustainable manner. It's probably one of the best examples in the world. Not going to say that it's perfect, but we do work very hard to try to make sure that those populations remain stable out there even while we extract those resources. So hopefully we're still doing a good job of making sure that those sea lions have plenty of pollock to eat too. But it is definitely one thing to be aware of and be concerned about, to make sure that we're not taking too much of the pollock. Now, does anybody know what an otolith is?
I already said.
It's a two-part word. So oto, does anybody know what that root comes from?
Light.
Oh, it's really close to photo. Oh, that's a good guess. Not correct, but it has to do with another one of our senses.
What was the word again?
Otolith. So let me see here. It's actually listed right down here in your paper. Otolith means ear stone. So oto is ear, and lith is kind of like lithography. That's a method of printing where you carve a stone and then use that as like a little rubber stamp almost. So lith is stone. So these are stones in the ears of the fish. And I'm actually going to take a break here from my scat and dissect a fish for a second so you can see where they come from. So I happen to have a pollock here. Handy, doesn't everybody have a pollock in their pocket? Let me get a little better lighting on my pollock here. So you can see pollock is not the prettiest fish in the world, but they are tasty. You don't normally see pollock straight up on the menu, but again, it's a type of cod. So it has 3 dorsal fins. 1, 2, 3. You know, this guy's pretty beaten up. It was packed into a giant block of many other frozen pollock, and so they don't look very shapely anymore once they've been in there. But it's just like other cod species, those 3 dorsal fins, and similar to the other cod, you know, it's a nice white meat on the inside. So I'll show you where the otoliths come from. I'm going to turn this around real quick. And right here on the operculum, the gill cover, I can see this little line. Right above that line, I am going to take my knife and just carve right down into the skull till I feel the skull break. And I'm just going to pop that open. And right there, in the center, right in these 2 little pockets next to the brain, are the otoliths. And you can see there's just these bright white things that just jump right out at you. They're not attached to anything. They're just floating free in these little pockets. And you can just slide them right out. So they're not part of the skeleton at all. And in the grand scheme of things, looking back at the size of this fish, these are pretty big rocks inside their heads. All fish, at least all the ones I know of, have otoliths. They help them with hearing, and they help them with their balance. So these are stones that they build using minerals from the water, and so you can use these otoliths for a variety of things. You guys could tell just by looking at yours that they came from walleye pollock. So you can tell what species they came from, and I'll show you another one from a different species. You can see a good example of how different the shapes are for different species. You could also tell by looking at the Pacific cod otoliths in your ID chart that cod and pollock look very similar. Again, that's because the pollock are a type of cod. But there's the walleye pollock otoliths, and these are otoliths from coho salmon or silver salmon. So you can just see the different shapes in them. And if you look closely at the texture of those otoliths, sometimes you'll be able to see the indication of little tiny rings. And these are growth rings, sort of like trees would have. It's the same kind of thing. So there's another piece of information you can get is from the fish itself, not just the species, but the age of the fish and the size of the fish as well. As if you look down right at the bottom of your worksheet, we have a little ruler, and you can use that to measure the size of your otolith. And in millimeters, if you collect that size in millimeters, mine's about 16 millimeters. Oops, way off the screen here, sorry. My otolith is not cooperating. So I've got a 16 millimeter otolith, and then if I flip over my worksheet, I can see how big that fish was that produced that otolith. So scientists have, of course, gone through and sampled a whole bunch of fish and measured the length of the fish and the length of the otolith, and then they used a best fit scenario to get that line through there so that we can predict or estimate about how big a fish was based on its otolith size. So my 16 millimeter otolith was a fish that was about 34 centimeters long overall. And then from that, I can estimate the weight of the fish. 34 centimeters was a fish that was about 0.9 pounds. Now, I have this in pounds, obviously not the unit that scientists would typically use, but when we talk about how big a fish is here in the U.S., we don't usually talk about how many grams my fish weighed. So it just means a little bit more to students when you say, I caught a fish that was less than 1 pound, you know, and had this huge rock that's bigger than my fingernail. It's a tiny little fish with this giant otolith in it. So these rocks in their heads are what allow them to hear sound. As the sound waves come through the fish's head, they bounce around in there. But they're particularly useful for scientists when we collect these things. These otoliths are even preserved in the fossil record, or just from sort of proto-historic times. We can find otoliths down in the sediment, and then from the chemicals that are in those otoliths, we can learn about the characteristics of the ocean at that time, because those fish were using the chemicals from the ocean. Those otoliths can even tell you a bit about ocean chemistry. And using isotope ratios, you can even find out during certain years of its life what part of the ocean did that fish live in, based on what we know about the differences in ocean chemistry in certain areas. So otoliths are extra valuable pieces of information. But let's get back to our scat samples and see what other kind of things you've identified in there. See if I can help you pick out anything. So what else did you find in your samples besides Pollock otoliths and the other fish bones that we identified?
Almost like a
What was that?
thing, but I'm not sure if it's like a vertebrae or if it's like a real fin support.
Oh, I got a bunch of those.
Yeah, I feel like it is.
Okay, you should have a lot of vertebrae, and most of the vertebrae will have lost the little spinous processes that go off the side, the little skinny parts, and that's just because through the digestion process, these fine little bones tend to just break apart. So most of them are just going to be the centra of the vertebrae. You should find a lot of those, and naturally, one fish, you know, might have 3 dozen vertebrae. So there's going to be a whole lot of those, and those won't tell you much about how many fish these sea lions are eating, but it can tell you a little bit about the size. You can see I've got a couple of really big ones and some smaller ones, and I might have some little teeny ones in my sample that I didn't find yet. So you can say something about the size, and there are some experts in the field that would be able to look at these vertebrae and actually tell you the species even. But that's a pretty tough one. You definitely have to be a fish anatomy expert for those. What about these parts over here? What are these? Those are eye lenses, and if you look at this big one in the middle, if I can stand it up, it's being really uncooperative today. If you look closely at that one, you can see how it's it looks like orange slices the way it's dried out. The one right next to it is just this clear ball, and then there's another one of those right over here. It's pretty clear. That actually looks like it almost has like a different kind of core inside of it. These 2 eye lenses are different, and probably this one too. These are from squid. So we've got some squid eye lenses in there as well, but this big one is a fish eye lens. So all these that sort of split apart like orange slices are the fish eye lenses, and these ones that look more like little pearls, little shiny balls, are squid eye lenses. So you can use those to estimate the numbers of fish or squid that your sea lion ate by saying, you know, if I find 2 eye lenses, that those both could have come from the same fish, or squid in this case. You can't really go by color necessarily, because as things digest, they can take on a new tint pretty easily. So these 2 eye lenses could have come from one squid, but I've got, let's see, 1, 2, 3, 4, 5, 6, 7 fish eye lenses. So how many fish did my sea lion have to eat to come up with 7 eye lenses? Had to have at least 4, right? It could have eaten 4 or 8 or 100 fish for all we know, because they don't poop out everything all at once, right? They don't just cleanse their whole body. But you can't come up with 7 eye lenses if you only have 3 fish or 2 fish. You have to have at least 4. So they can say that they at least ate that many. With otoliths, it's similar because there's a left and a right otolith. You can tell the difference between the left and the right side, if you know what you're looking for. And also the size, you know, this one's vastly larger than these others. So these 2 might have been from the same animal here. This is one animal. These two could have been the same animal, but then I've got another piece that may or may not have broken off of this one. Looks like it came from another animal entirely. But they can sort of pair those up. And if these four were all from the same side of the body, and it looks like these two were, then I could say that's 4 different fish. I have 4 left otoliths, for example. So that's a way for scientists to get some semi-quantitative information from these scat samples. Now, the true quantitative data would come from like the length of the otolith. They can say how big a fish these animals are eating. Does anybody know what these things are here? You may or may not have found any of these in your scat sample. Let me get a piece of paper under there. What are those? Minus the vertebra.
I've got some stuff that kind of looks like that.
Anybody know what they are? This must not be the same class that you had last year, Crystal.
Yeah.
Didn't you dissect some squid with some of your students?
Yeah, last semester.
Yeah, okay, that's what I thought. So these are squid beaks. And again, like the otoliths and the eye lenses, these come in pairs. There's a top and a bottom to a squid beak. And so there'd be 2 halves. So you could use these to get some more quantitative information about how many squid these animals are eating. There might be some size relationships to these. Those are a little tougher to create, but scientists do have some information about the size of a squid's beak versus the size of its body. And from that, then you can say how many calories they're getting from certain prey types. How about these here? These clear, round, clear things, flat. What are they? They look totally clear, just a little bit like a little plastic disc. And they're found all over the outside of a fish. Those are the scales, and in scales you can find little growth rings, again, kind of like the otoliths. Which brings me to a very interesting concept, I think, here in Alaska. With the salmon fishery. So I'm going to show you an image of a salmon otolith, and I want you to tell me what sort of information you could glean from this. So this is under a microscope. These are microns, these measurements here. And looking closely, do you see anything that seems unusual or interesting? Or what do you think happened to create this image? This is inside the center of the otolith. All right, I'm going to give you some clues. I'm going to point out there are 4 lines that are very evenly spaced, and then a small gap, and then 3 more very evenly spaced lines. And I'll give you one more clue. Right here is when this salmon hatched from its egg. This is called the hatch line. That's its first period of growth after it hatched from an egg. So all of this happened before this egg hatched, these 4 lines and these 3 lines. What do you think caused those lines to appear in this otolith before it hatched?
Stress?
A salmon egg is about pinky nail sized.
Not stress.
I couldn't quite hear you. Say it again.
Stress? Sir?
Stress. Well, that's a good thought. Yep, because that would change its growth rate potentially. I'll give you one more clue. The main factor that influences growth rate, or development rate, I should say, in a salmon while it's still in the egg phase is temperature. So the temperature went basically up and down 4 times in a row, and then it waited for a second, and then did it 3 times in a row again over the course of this egg's development. What do you think caused those lines, or what do you think caused that temperature to change like that?
Seasons, storms, seasons, rainfall.
Seasons, rainfall. This all probably happened over the course of, I don't know the exact number of days. Actually, I probably have the data right here. Over the course of about 16 days. This was artificially induced. So these were in a tank, in a hatchery, and they increased the temperature from 5 degrees C to 9 degrees C 4 times in a row, just one day, on, off, on, off, on, off, on, off, and then waited 1 extra day, and then 3 more times, and they basically put a little barcode inside the head of this fish. So this was a hatchery-raised salmon, and the code that we put into it was 43H. That says 4 lines, 3 lines, hatch. So when a scientist collects this otolith, after the fish has been caught, they can look inside of it and see the code 43H. They can go to the Alaska Department of Fish and Game website and find out that this was a Chinook salmon, king salmon, released from the Alaska SeaLife Center in 2005, and it was from that population right there, released in the Seward Lagoon from our population. So this is a way for hatcheries to mark millions of fish, all of them, just by ramping the temperature up and down. So there's more information that can be collected from otoliths. Even through the hatcheries, it's a way for us to track those fish. Of course, at the far end, it requires you to collect that otolith and then grind it down, look at it under a microscope. But there's a lot of detailed information inside those otoliths. They're probably the single most valuable piece of information that we can find in these scat samples, because they're so easy to use to tell the species of fish that the sea lions have been eating and the sizes of fish. But there is a lot more out there, including those hormone levels. So one thing, I don't think any of you found it because you probably would have said something, but these days we actually feed the female sea lions different colors of glitter so that when we collect their scat samples fresh, we know who it came from, and we can test those hormone samples when we get the opportunity. So we collect that sample. We don't know who it came from except by looking at those markers, the different colors of glitter that we use to keep track of whose poop is whose here at the SeaLife Center, which makes it really funny when we're diving in the tank and going and sucking up the poop off the bottom. There's all these little red sparkly things down there in gold and silver. That's really fun. All right. I think you guys need to get going, but I'm happy to answer any last questions that you have before you need to take off.
So when you examine the otoliths like that, that can only happen in captivity because, so like in nature, wouldn't you see like, because the temperature changes, like as nighttime would fall, so you would see like 16 of them, essentially.
It wouldn't be such a drastic change in temperature probably with a natural an animal that was just raised in a typical stream, because streams are flowing. The temperature doesn't go up and down that much. But something that we know with salmon in particular, there's been so much research done. You know exactly how many acquired thermal units it takes to hatch them. And so they can put those codes in there, and they know exactly when that egg is going to hatch based on the temperature. But so they can ramp it up quite a bit more than would be acceptable in the wild. There's a temperature range out there in nature that would actually be disastrous to, or if a temperature threshold, if it gets too warm, that would be disastrous to those little developing eggs. But it's typically going to be a much narrower field of temperature in that stream in the wild. They're laid in little gravel beds in pretty steadily flowing water, if not very fast-moving water even. So yeah, the temperature just doesn't go up and down that much.
So it would be a lot harder to identify than wild.
Yes, for a wild, you would be able to tell just by looking at the otolith. You'd be able to tell that it was not marked for sure right away. And I'm sure there are other things that they can look at to detect that it's a wild salmon otolith, because they don't necessarily mark all of the hatchery fish. But there is probably some kind of subtle differences between the wild and the hatchery-raised ones within those otoliths. But they'll also do things like clipped fins, so that on the exterior of the fish, as it's swimming up, you can see that it's missing a fin. That's really a quick, easy way to detect that it's a hatchery-raised fish. But that requires you to, with these fish that are just 1.5 inches long, go and snip them off with little tiny scissors, So you can do it. They do it with tens of thousands of them, but it's a whole lot more work than just raising the temperature up and down while you're raising all of those little eggs. So they've got some other tracking methods for salmon too. They'll even put acoustic tags in some fish so that you've got this little passive acoustic receiver that just detects when one of them swims by. And they've got a lot of neat technology out there for salmon. It's a huge deal for Alaska, of course. What I think is fun is when we're trying to catch those salmon, the stellar sea lions are usually just a few feet farther than we can cast. And they're out there shaking them around and swallowing them whole just beyond where we can fish. So they do a much better job than we do of catching those animals. They are very effective predators.
You guys,
All right, any other questions?
Darren, thank you so much for time out of your schedule. I really appreciate it. Everybody else give a big applause for today.
You are very welcome. I'm glad we got the technology working well enough anyway. And if you guys get the chance to come visit us here in Alaska, definitely come check it out. It is an awesome place. You can get a summer job and just hang out for the summer.
Thank you again, Darren. I really appreciate it. Oh, and tell Caitlin I said hi for me if you could.
All right, I will.
Okay, thanks.
All right,
Bye now.
take care. Have a great afternoon. Bye-bye.